Hello friends, welcome back to my YouTube channel Nesting Padasala. In this video, I'll be sharing you 10 questions which you can expect for GATE Life Sciences Microbiology paper. First is match the following type of questions where they will be asking you to match the scientist with their contribution. First scientist is Sergei N. Vinogradsky who had discovered nitrogen fixing bacteria in soil. Next is Louis Pasteur who had established germ theory of disease, and Joseph Lister who had developed aseptic techniques, and Edward Jenner who had developed a vaccination for smallpox. Louis Pasteur is called as father of microbiology, Robert Koch is called as father of bacteriology, Joseph Lister is called as father of antiseptic surgery, and Edward Jenner is called as father of immunology. And these are the respective contribution of Louis Pasteur. He had proposed the principle of fermentation, the principle of sterilization, and he was the one who had developed autoclave, and he had described the method of pasteurization, and he had contributed for vaccine development for anthrax, cholera, and rabies, and he was the one who had postulated germ theory of disease. And Robert Koch, he had introduced soil media, and he was the one who gave pure culture technique and hanging drop method, and he was the one who discovered bacteria causing anthrax, cholera, and tubercle, and he also introduced a staining technique. Next, Joseph Lister was the one who identified like post-operative infection can be reduced by using this 5% of carbolic acid solution. And Edward Jenner, who gave a first vaccine called smallpox vaccine, he had used a cowpox virus who immunized the children affected with smallpox. Next, flagella arrangement. So bacteria having clusters of flagella at both the poles of cells are known as, it is known as Lophotrichus. So already we discussed monotrichous bacteria will be having a single flagellum. Example include cholera vibrio and the Pseudomonas aeruginosa. Next is Lophotrichus where they will be having a several flagella which is also called as tufts of flagella which will be extending either from one end or both end of the cell. Example include Pseudomonas fluorescens or spirilla. Next is peritrichus where the multiple flagella will be randomly distributed around the entire bacterial cell. Example include typhoid causing bacillus. Next is amphitrichus where the single flagella or multiple flagella that will be extending from both ends of cell. Example include alkaligenes spicalis. The third question is which of the following disease for which a chemotherapeutic agent? First chemotherapeutic agent or use. The answer is syphilis. So of course earlier the first successful chemotherapy was with regarding malaria where Indian used the bark of cinchona to treat malaria. But here they are asking the first chemotherapeutic agent. The first chemotherapeutic agent was given by the work of Paul Ehrlich. Okay, he had discovered two drugs. One is para-rosanilin, which is having an anti-trypanosomal effect for treating trypanosoma, and next is arsenicamide, which is also called as salvarsan or compound 606, which is used for treating syphilis. Okay, and he also postulated like it would be possible to find few chemical, like it is an organoarsenical compound. It is an arsenium containing compound and it is selectively toxic only for parasite, but it is not toxic for human. Okay, so this idea has been called as magic bullet. Okay, these are the other contribution of Paul. So he had discovered the acid for staining technique, and he was the one who discovered the minimal lethal dose, and he was the one who proposed sidechain theory of antibody production, and he done his immense work in the field of chemotherapy and immunology. Which of the following agent irreversibly oxidize and inactivate the protein with XSH group? The correct answer is iodine. So these are a list of other disinfecting agent and mechanism of action. Mechanism of action of alcohol include denaturation of protein, whereas chlorine, they oxidize the sulfur hydryl enzyme and amino acid. At the same time, they will oxidize the respiratory chain compound, and they will be decrease the oxygen uptake, and they will be decrease ATP production. At the same time, they will be inhibiting the protein synthesis, and they will be inhibiting the DNA synthesis by breaking the structure of DNA. So formaldehyde, they alkylates the amino group and the SH group of protein. Next is at the same time they also alkylate the ring nitrogen atom of purine. Glutaldehyde which alkylates SH group, OH group, carboxyl group and amino group of protein. So both this formaldehyde and glutaldehyde, they are highly reactive compound which will be readily combining with the organic N compound present in nucleic acid and protein and inactivate them by cross-linking and alkylating. Okay. Next is hydrogen peroxide. They will be producing destructive hydroxyl free radical which will be attacking the membrane lipid, DNA and other essential cellular component. Next is iodine. As I mentioned you earlier, they are oxidizing agent which will be irreversibly oxidizing. At the same time, they will be inactivating the protein which SH group. The next role is that they will be causing the halogenation of those protein with or those enzyme with tyrosine unit. Okay, so they will be attacking by halogenating the cellular component that require tyrosine for their activity. So the next disinfectant is phenolics. So at high concentration they will be acting as a protoplasmic poison and they will be penetrating the cell wall and they will be disrupting the cell wall component. At the same time they will be precipitating the cellular component. Okay, so cellular protein. Next, a low concentration of phenol or when a high molecular weight phenol derivative has been used mean it will be cause of bacterial death by inactivating the essential enzyme system. At the same time they will be causing the leakage of essential metabolite from the cell wall. Okay, the last disinfectant is quaternary ammonium compound which will be inactivating the energy producing enzyme. They will be denaturing the essential cellular protein and they will be disrupting the cell membrane. Next, there will be definitely one question coming from the disease and the causative agent. So here I had properly matched the itself. Okay, so Q fever, which is also called as query fever, is caused by Coxiella burnetii. Plague is caused by Yersinia pestis and rabies is caused by Lysa fever, Lysa virus and malaria is caused by Plasmodium. So here I had given you the pathogen, the vector, the genus name of vector. So yellow fever, Japanese encephalitis, West Nile fever, dengue, Zika and chikungunya are caused by flavivirus and all are transmitted by the mosquito only. If you take yellow fever, Japanese encephalitis, West Nile and dengue, Zika, it is caused by the mosquito belonging to genus of Aedes and Culex. Whereas if you take chikungunya, it is transmitted by mosquito genus of Anopheles and Aedes. So next is bacteria. So Yersinia pestis is caused by plague and the vector is flea belonging to genus of Xenopsilla. Okay. Next is Rickettsia provaxi which is caused typhus and it is transmitted by lice of genus Pediculus cumanus. Next is Borrelia which is responsible for causing the Lyme's disease and it is transmitted by ticks belonging to genus name of Dermacentum. Next is protozoan. So first protozoan is Plasmodium which is causing malaria and it is transmitted by mosquito belonging to genus of only Anopheles genus of mosquito alone responsible for causing malaria. Okay. Next is Trypanosoma. So Trypanosoma cruzi is responsible for causing Chagas disease and the transmitting vector is kissing bug of genus Rhodinus striatoma. Okay, so these two genus alone are responsible for causing the transmitting this particular disease. Next is Trypanosoma brucei which is responsible for causing sleeping sickness and it is transmitted by tsetse fly of genus Glossina marcitans. Okay, this is a scientific name because these time scientific name is also been asking. That's the reason I had given you. Leishmania is responsible for causing leishmaniasis and it is transmitted by S fly belonging to genus of these two genus. Okay. Next is filarial worm. This include Onchocerca river blindness. So river blindness is a parasitic disease which is caused by the genus of which is transmitted by the black fly belonging to genus of Simulum. Okay. So this particular is called as river blindness because generally this particular fly which is responsible for transmitting this particular disease, they generally breed near the river, fast flowing river and lakes. That's why it's called as river blindness and it is caused by the filarial worm. It is transmitted by the filarial worm called as Onchocerca volvulus. Okay, and it is transmitted by the bite of this particular fly belonging to this particular genus called Simulium. Next is this particular disease which is called as loa, which is called as worm and it is also called as African eyeworm disease because this particular disease is present in the conjunctiva, that is it is transmitted by the deer fly which is also called as mango fly or mangrove forest fly belong to genus of Chrysoprus. Okay, and this disease is transmitted by the repeated bite of the deer fly. Okay, so it is also called as African eyeworm disease because the filarial worm will be present in the conjunctiva of the eye. Okay, this two particular filarial worm of filaria, that is Ucheria branchofti or this particular thing Bergia malai, sorry if I pronounce the scientific name wrong. These two filarial worms are responsible for causing lymphatic filariasis and it is transmitted by mosquito belonging to genus name of all the 4 species of mosquitoes responsible for causing this particular disease. Next question is resident macrophages of DAS are called as Kupffer cell. So resident macrophages of liver is called as Kupffer cell. So here I had given you resident macrophages of adipose tissue include called as adipocyte, adipose associated macrophage and it is responsible for controlling the insulin sensitivity. In blood you can able to find monocyte which are a precursor of macrophages and they are acting as a intravascular housekeeper. The main role of this resident macrophages of blood is that they will be clearing the endothelial cell debris. Next in bone you can find osteoclast osteoclast cell, at the same time bone marrow macrophages. Okay, so osteoclast cells are the resident macrophages present in bone. Next, central nervous system has three resident macrophages. So the perivascular and meningeal macrophages are responsible for immune surveillance, and this microglial cell are responsible for the removal of dead neuron. At the same time, they will be promoting the neuronal survival. Next, in gastrointestinal tract, the resident macrophages is intestinal macrophages, it is called, and it is responsible for regulating the immune response to commensal bacteria. At the same time, they will be maintaining the intestinal homeostasis. Okay. Next, in liver, the resident macrophages is called as Kupffer cell, where they are responsible for clearing the microorganism and the cellular debris from the blood. Okay, at the same time, they also clear aged erythrocyte, and the next macrophages is called as motile liver macrophages, which is responsible for immune surveillance. And in lung, the macrophages, resident macrophages is called as alveolar macrophages, which is responsible for immune surveillance of lung for the inhaled pathogen. Next, in skin, you can able to find dermal macrophages and Langerhans cell. So both are responsible for immune surveillance only. Okay, so in spleen you can able to find four macrophages. For first is marginal zone macrophages and metallophilic macrophages, which are responsible for immune surveillance. This red pulp macrophages is responsible for erythrocyte clearance and iron metabolism. Next is white pulp macrophages present in spleen are responsible for clearing your apoptic cells. Okay, next the fibromouracil is a base analog of which base? So it is a fibromouracil is a base analog of thymine. Okay, so it causes transition mutation. So this fibromouracil mispairs with guanine instead of pairing with adenine. Okay, so here I have given you alkylating agent includes ethyl methyl sulfonate, methyl methane sulfonate, ethyl ethane sulfonate, and ethylene imine. So ethyl methyl sulfonate and methyl methane sulfonate causes AT to GC transition, whereas ethyl ethane sulfonate causes GC to AT transition. Next, base analog include 5-bromouracil and 2-aminopurine. So already I discussed 5-bromouracil. This 2-aminopurine is a base analog of adenine, which will be mispairing with a cytosine instead of thymine. Okay. Next is acridine dyes like acriflavine or proflavine. They will be acting as an interchelating agent, and they will be acting as an interchelator, and they will be responsible for deletion mutation, addition mutation, and friendship mutation. Other include deaminating agent like nitrous acid, which will be a deaminating agent, and it will be deaminating the cytosine to adenine. Okay. Next is hydroxylamide and sodium acid. Okay. Next, which of the following combination is correct? So, penicillin inhibition of cell wall synthesis is correct. Rest of the thing are wrong. So, here I had given you correct thing. Cell wall synthesis inhibitor include vancomycin, bacitracin, penicillin, cephalosporin, and these things. And folic acid metabolism inhibitor include your trimethoprim and sulfonamide. So both the sulfonamides and trimethoprim are the example of antimetabolites that will be interfering the bacterial synthesis of folic acid by blocking both the purine synthesis and pyrimidine biosynthesis, thereby inhibiting the bacterial group. Okay, whereas this two bacteria like polymyxin and daptomycin, they generally cause breakdown of your cellular membrane structure, and they will be inhibiting the function of cellular membrane. Okay. Next is with regarding to this quinolines, which will be inhibiting DNA gyrase enzyme activity, and again they will be inhibiting the process of DNA replication. Next is actinomycin, rifamycin, and the streptoviricin. They inhibit the process of transcription. This actinomycin inhibit RNA elongation, whereas this rifamycin will be inhibiting the activity of DNA directed RNA polymerase. Next, the antibiotic that inhibiting the protein synthesis, that is these antibiotic like erythromycin, chloramphenicol, lincomycin, they are called as 50S inhibitor. Okay, so they will be blocking or inhibiting the step of initiation of transcription or elongation of transcription. Sorry, initiation of translation or elongation of translation. Next, 30S subunit inhibitor include tetracycline, pectinomycin, streptomycin, gentamicin, kanamycin, and nitrofurans. So they will be blocking the access of amino acyl tRNA into the process of translation. Okay. So the last thing is puromycin, which will again inhibit the process of translation, but this puromycin will be inactivating or it will be disturbing the structure or function of the transfer RNA. Okay. So here I had given you So this actinomycin D is an anti-neoplastic agent that will be inhibiting the RNA synthesis by binding to G residues. At the same time, they will be inhibiting the DNA different RNA polymerase. These are the additional references like inhibitor of bacterial cell wall synthesis against subdivider. So the cycloserine, which will be inhibiting biosynthetic enzyme responsible for cell wall synthesis, whereas this bacitracin, they will be combined with a carrier molecule and they will be inhibiting the function, and this vancomycin will be combining with the cell wall substrate. And next, the drugs like penicillin, cephalosporin, monobactams, they will be inhibiting the polymerization and attachment of new peptidoglycan to the cell wall. Okay. Next is inhibitor of cytoplasmic membrane like this polymyxin, they will be disorganizing the cytoplasmic membrane, whereas this gramicidin, they will be producing pores in the cytoplasmic membrane. Next is this imidazole, they will be altering the structure of the cell membrane of fungus. Okay. Next, these are the inhibitor of nucleic acid synthesis. So first inhibitor of nucleic acid metabolism, nucleotide metabolism include this adenosine arabinoside in case of viruses, acyclovir in case of virus, and flucytosine in case of fungi. Okay, so the next, the agent that impact DNA template function include the intercalating agent and chloroquine in case of parasite. Next is inhibitor of DNA replication include quinoline and nitroimidazole. Next is inhibitor of RNA polymerase include rifampicin and actinomycin. So these are the inhibitor of 30 subunit and these are the inhibitor of 50 subunit, and here I have given you inhibitor of folate metabolism. Okay, so trimethoprim is an inhibitor of dihydrofluoride reductase, whereas the sulfonamides are the inhibitor of petroic acid synthesis. Okay, synthase inhibitor. Okay, next is with regarding to Anton von Leeuwenhoek observed several microscopic organisms under his handmade microscope. He described them as animalcules. Okay, so these are the contribution of Anton von Leeuwenhoek. He was the one who had constructed the first biconcave microscope, and he was the first one to observe microorganisms from water, mud, saliva, and intestinal contents of healthy subject. And he was the one who gave the name for animalcules. Okay, he named them as animalcules, and he was the one who done an accurate description of bacteria like cocci, bacilli, spirochetes. Okay, and next we will be studying about Koch postulate. Okay, so Koch has given 4 postulates. First postulate is that organism should be regularly found in the lesions of the disease. Second postulate is that it should be possible to isolate the organism in pure culture. Okay, and he was the one who gave pure culture technique as we saw them earlier. Next, postulate number 3 include inoculation of pure culture into suitable laboratory animal should reproduce the lesion of the disease. Okay, the last postulate is that it should be possible to re-isolate the organism in the pure culture from the lesion. Okay, so these are the 4 postulates, and the virus and microbacterium doesn't fulfill the Koch postulate. The reason is that virus cannot able to grow on any kind of artificial media. At the same time, this particular microbacterial leprae, which is responsible for causing leprosy, can also, we can't able to culture them in an artificial media. Okay, next there will be one question asked from gram staining. So in gram staining, iodine is used as a mordant, which means it is used as a dye fixative. So this gram iodine will be combining with the crystal violet and they will be forming a large complex which help them to adhere to the cell wall of the bacteria. Okay, so this in gram staining, gram iodine is used as a mordant. The primary staining is crystal violet and the counter staining is your safranin, and decolorizing agent is your ethanol. So here I had given gram staining is used, it uses crystal violet as a primary stain, gram staining as a mordant, ethanol as a decolorization, and safranin as a counter stain or secondary stain. It is generally used to distinguish the cell wall of gram positive and gram negative. So gram positive cell will be staining purple or violet and gram negative will be pink in color. Next is acid fast staining technique, which is for using acid fast bacteria like your microbacterium species, where the cell wall will be having a high amount of lipid or mycolic acid content. So we can't able to use a usual crystal violet staining technique and all. So that's why they have given this acid fast staining, where they will be using 2 dye. One is basic fission and next is acid alcohol. So this acid fast bacteria, they will be resist the decolorization by acid alcohol. So they don't leave the color. Whereas this non-acid fast bacteria can readily decolorize by this acid alcohol and they will be taking a counter stain of methylene blue. Okay, so acid fast bacteria will be staining red in color and non-acid fast bacteria will be staining blue in color. Next is endospore staining, where they will be using they will be using a heat, and next they will be using a malachite green, next they will be using a counter stain saffron. Okay, and it is used to distinguish organism with endosperm and without endosperm, and also used to study endosperm. So endosperm will be appearing bluish green in color, and other vegetative structure will be appearing in pink color or red color. Next is flagella staining. So it is used to study about flagella types and other flagella. So for flagella staining, they will be using the flagella will be coated with the tannic acid or potassium alum, which is used as a mordant, and then they will be using a counterstain called as pararosilin or basic fusion, and it is used to study the flagella. Okay, next is capsule staining, which will be using, which is also called as negative staining technique, and in this capsule staining generally used to study or observe capsule containing bacteria. Okay, and it is used to distinguish cells with capsule and those bacteria cell without capsule. Okay, the dye used include India ink or Negrosin. Okay, so the counter stain is also being used. So capsule staining is used to study about capsule containing bacteria, and these are the list of important 10 questions that you can expect for GATE microbiology paper. Thank you friends, thanks for watching this video.